Good morning, everyone. In Rory's opening remarks, he mentioned the baseline assessment process where we collect data, physical measures and samples. And I'm going to spend the next 10 or 15 minutes telling you about the samples that were collected, how they were processed and how they're stored and how they could be made available to people. Before I do that, I just want to spend a little bit of time describing some of the thinking that went in to the sample collection protocol. Because when we started this process in 2003, we had two challenges. And the first of those, of course, if you're doing anything on 500,000 people, you can't go back and do it again. And the second thing is, of course, with this being a prospective study, we want to collect samples that, as far as we're able, will be most useful to researchers in the future. And of course, that's very difficult. It's very difficult to predict technology trends in 3 to 5 years, let alone in 15 or 20 years over the lifetime of the study. So as with a lot of the areas of UK Biobank, an expert advisory group was set up under Paul Elliott from Imperial College to advise us on how to go about this. And I'm just going to summarise the approach to that because it's quite informative. Clearly, the overarching philosophy was that the baseline and samples from repeat assessments should give an accurate representation of the biology of the individual at the time of collection. And that group developed a set of clear criteria for inclusion, which samples should be included, which sample types should be included, and also those which should be excluded. And we settled on peripheral blood, a spot urine, a midstream urine, and in the later phase, the enhancement phase, a mixed saliva sample. The approach taken was that the protocol had a strong scientific lead, and that was the foremost factor. But of course, on something of this scale, we had to consider cost and also future-proofing, not collecting and processing samples in such a way that they may preclude future analysis as far as that could be predicted, and collecting them in a way that would allow as broad a range of analyses as we could foresee. We had to consider the consequences of scale in this, in how these samples would be collected and processed, and we thought very hard about whether they should be processed locally or centrally, with the strengths and weaknesses of both of those approaches. Having settled on approach, we had to show that the samples that we were collecting and processing were fit for their intended purpose. There were lots of papers in the literature showing the impact of particular processing protocols on different analytes under different conditions, but there wasn't really a coherent summary that was suitable for UK Biobank, and this was work that Paul and the group advised us on. And then the last sort of operating principle, if you like, is that the analysis of the samples at the time of collection would be minimised, and minimised to those analyses that could only be done on fresh sample. And that, as you'll see, was a series of haematological analyses. Because at the time, of course, we didn't know which assays would be the most informative for people. And also, if we could defer those to the future, when new technologies would be available or at much reduced cost, that would be advantageous. And we've seen that with the genotyping that you'll hear about later in the day. We knew that perhaps the biggest source of variation would be the so-called pre-analytical variables. What happens to the sample between the collection from the individual to the point where it's cryopreserved and stabilized? So a lot of work was done looking at things like the selection of anticoagulants. If you take, for example, heparin, heparin will interfere with some functional assays, it will bind to proteins, and it may interfere with some DNA-based assays. But we were advised that if you want to do metabonomics in the plasma, then we should use a heparin-based tube. EDTA, with its chelation properties, will also interfere with some functional assays, but gives very good DNA. It's a very good general source of DNA and plasma for biomarkers. Similarly, with the clot accelerators, the gold standard was to use thrombin, and this is where we come back to the point about cost. Doing that on 500,000 people would have been very expensive indeed. We were concerned if we were going to delay the processing of these samples from when they were collected, that hemolysis may have an effect on downstream assays such as homocysteine or potassium, so we had to consider that. And actually the control of clotting at the time, there were papers that suggested, and this has been reinforced since, that the proteome profile of the serum changes over the clotting time, so we needed to control that and record it for researchers. There was a lot of discussion about the use of preservatives and stabilising agents within this concept of future-proofing and trying to modify the samples as little as possible. RNase inhibitors, for example, will have, well, we don't know what effect that will have on other analytes in the samples, but we do, as you'll see, have a separate tube for the collection of RNA. We could have added a protease inhibitor cocktail, and that certainly would have inhibited protease activity, but we had no idea really what that would do to other analytes. And you can see we went down and considered all these various factors very, very carefully. It shows you how the thinking has moved on, because at the time we were concerned we were going to take a Buffy-coat fraction from the EDTA, but we were concerned that we would have insufficient DNA for future analyses, and we would need to have an inexhaustible supply of DNA. And the way that we would do that is we'd freeze cells down that in future could be immortalized using Epstein-Barr virus, and that would give us an effectively inexhaustible supply of DNA. So we thought very carefully about which cryoprotectants we should use. Clearly the processing time to minimize as much as possible, and the control of the temperature was important. What temperature these samples should be archived at, and what steps we could put in place to minimize the impact of freeze-thaw cycles on these samples. That was a great deal of work. That was about 2 years' work led by that group, and we came up with the following sample collection, and these are the samples which are available to you now. And this is in phase 1, and you can see there are 7 tubes collected. The small purple cap tube 6 in, that was the one that was used for the hematological analysis, and all the rest you can see underneath. They're EDTA tubes that give us plasma, Buffy-coat and red cells, serum and plasma, and so forth. You'll also note the orange-capped and the green-capped tube have an inert gel plug in them. What happens here is this is the only processing that happens at the assessment centre, is they're centrifuged, and that forms a barrier between the cellular component and the plasma and serum, and protects the samples from the effects of hemolysis. All of the tubes are barcoded, which gives a very, very robust and secure link between a unique identifier for the participant and all of the downstream processes. And this really is the first point in the sample processing where we started to build in quality assurance rather than detect problems at the end. So the barcode gives you a very, very rigorous link all the way through. It allows you to record time and location of collection. And the first 3 digits in that barcode identify the tube type to the robotics that process it. So you can't put the wrong tube type on the wrong robot. Subsequently, when we received this additional enhancements funding that Rory mentioned in his opening remarks, we added 2 additional tubes, a saliva tube, which is just a 20 ml Falcon tube, and a Tempest tube there, which just contains an agent which lyses the cells and stabilizes nucleic acids so that we can extract RNA from those tubes. There was a fractionation protocol developed so that we could, you can see here, we can get all these different fractions from these tubes, and then clearly we wanted to isolate those fractions, but also then sub-aliquot them to try and manage the impact of freeze-thaw. And then these were split between a working archive where the samples are held at -80, and I'll describe this, and a nitrogen vapor archive which was geographically separated. At the end of this process, in phase 2, where we have the additional tubes, we produce 36 aliquots from each individual. And as I mentioned, these are stored in two archives. The -80 is the working archive where the samples will be issued from because it's fully automated. And as you can see, they're more or less mirrored. So the geographical separation gives us the protection of a catastrophic event at one site. But also, all the literature tells us that -80 is sufficiently cold to stabilize all the analytes. If it turns out that's not true in the future, we can go to the backup sample in most cases in the nitrogen vapor, which is at -196. So coming back to the process demands of this is where we started to think, well, if we're going to follow this through, we need to be able to process these samples. And as you'll see, this required a slightly different approach. Rory described how we had typically 6 or 7 assessment centres open each day, each recruiting 100 people. So that gave us 700 participants per day. And in phase 2, that's over 6,000 vacutainers of primary sample, which produced 25,000 aliquots of sample each day. And they're stored in these small 1.2 ml cryotubes with a 2D barcode on the bottom. And by the end of this study, we had 15 million aliquots. Now, just because that's big doesn't make it good. And what we have to remember is what we're trying to do is produce a reliable process that will provide fit-for-purpose samples for researchers, both storing them and retrieving them. So in the black box, that is the what. The thing that I showed you at the start is the how. And if you come to UK Biobank, you'll see lots of automation. It's only like that because it's the only way we can do this in a high-throughput, high-quality, controlled and secure way. We thought very hard, as I mentioned, about whether we should process these samples centrally and locally. And I would emphasize, this is a consideration that if you're running your own study, the answer will vary. There's no right or wrong answer. But clearly with local processing, the processing is as rapid as you can really make it. So you'll preserve most species, even the labile ones. But at the time, we were very concerned. If you think about 6 distributed centres with these kinds of numbers, the quality control and the quality assurance of manual processing at this scale was a real concern. And bear in mind, you're using these samples in comparative studies, so you're looking for consistency. The robustness of the data trail is a concern, the risk to locally stored samples, because you can't put in place all of the infrastructure you can put in place with a central store to protect these samples. For us, and I say this will vary between studies, the cost of doing it locally was about twice that of doing it centrally. Of course, if you do it centrally, there is a delay. These samples were collected and shipped overnight and then processed. And of course, that increases your shipping costs quite substantially. But it gave us a much increased quality and consistency in processing and in our processes as well. We got a very robust data trail, and we were able to implement a lot of quality procedures to try and make these samples in as good condition as possible. Having made that decision, we thought we'd better be certain that that was right. We all felt that it probably was the right decision, but we felt we'd better check this. So we did a conceptually quite simple comparative pilot where we took a set of samples from 40 people in the age range, 20 men and 20 women. We took samples and processed them immediately, in other words, T equals 0, and then we held these samples at 4 degrees C for different times and then did a comparative study. And you can see we went through a very extensive range of analysis. All of these data are published in the International Journal of Epidemiology, and what they show is the great majority of samples are stable and fit for purpose. For some samples, there's a very small degradation. For 1 or 2, such as carbonate, it's quite a large degradation. And we're just in the process of publishing a paper with Paul Burton to show how you can compensate for those rare occasions where there is some degradation. So I draw your mind back to that slide where I showed you we were doing 700 people, 6,300 vacutainers, and 25,000 aliquots and so forth. What that actually needs is a slightly different approach to the way that you might do this on a smaller scale. And this slide here shows, to me, one of the key success factors for UK Biobank in that we were able to merge the academic elements with the process disciplines. And we recognised quite early that the study could be split into 3 sections. There's the design of the study and the protocols, which is an academic endeavour. And then the use of the samples and the data and the subsequent analysis is also an academic exercise. But the bit in the middle really is a process. And the more standardized you can make that, the more consistent you will make your sample collection. And of course, it includes things like sample archiving and distribution and processing, but it also includes all this other stuff at the bottom, which may not be the most exciting thing for a meeting like this, but it is rather important to make something on this scale work well. And this was a really important aspect for UK Biobank. As a consequence, the UK Biobank facilities perhaps don't look like a typical academic centre. They are, in fact, just in this light industrial unit on the south of Manchester. You see it the top left as you look at it. And the sample processing area, bottom left, just has lots of large-scale automation in it to enable us to do this at high quality and high throughput. And then on the right-hand side, you can see the -80 automated archive, and I'll talk about this a little bit more in a moment. You can see in there, they're just in these big, open, steel-framed buildings with a very level floor, and that's really all it is. They're very, very flexible buildings and quite a cost-effective way of doing it. I mentioned at the start that we tried to build process into the quality, and using the automation enabled us to do this. This image here shows one of the high-throughput sample processing robots. It's based around these anthropomorphic arms, industrial scale, very, very standard, just workhorses that did all of the sample processing. And the only really sort of interesting bit in this was the system put in place to deal with the heterogeneity that you will find in the population in these different samples. So these pictures here show 2 EDTA tubes which have been centrifuged, and you can see the interface. You can see this heterogeneity in the relative volumes here. On the left-hand side, as you look at it, these tubes have been photographed against a datum point in the middle, backlit, and you can see the interface between the cellular component and, in this case, the plasma very, very clearly. From this tube, we also wanted Buffy coat. So on the right, you can see a picture where it was photographed, where it was lit from above, and you can see the buffy coat, that white line, very, very clearly. And the automated system allowed these images to be overlaid, and because it's photographed against a datum point and you know the geometry of the tube, you can work out the heights of these things for all of each individual tube, and then those heights are associated with a barcode and converted into a set of pipetting instructions. So we got very good quality, and all of this was done at 4 degrees C. And we were able to test this with very fatty plasma, because this is a system based on a camera. Of course, you need to be able to distinguish this where people have had a very fatty meal or they have perhaps a hyperlipidemia. And using these systems allowed us to get the average processing time for all of our samples to 24 hours, and to make sure that that processing time was nice and tight. We didn't have big long tails on our processing times. The right-hand side, as you look at it, shows the inside of the -80 store. And this holds 10 million samples on these trays behind these. You can see these sort of blue insulating bricks. The samples are held behind there at -80. And this big yellow R robot will either put them away during the collection phase or retrieve them when researchers need them. And it kind of looks big and it's very impressive. It only does 3 important things. It keeps the samples at a very, very even cold temperature. Top left, you can see some experiments where thermocouples have been placed around these different quadrants and the temperature measured. You can see it's very, very stable and level, and you want that to keep your samples in good condition. Even when you open the drawer up, the space in the middle where you can see the robot is at -20 because large-scale automation won't work at -80. You can see at the bottom right, the pink trace that goes up to about -15 is the air temperature when you open one of these drawers. The yellow trace is a thermocouple placed in a tube which is in one of the trays which is opened, and that only goes up to -65 and then it slowly comes back down. All of the adjacent drawers, the temperature is unaffected. So that's the first thing it does. The second thing it does, it keeps it in very good condition in that it prevents frost build upon the samples. The typical humidity in the air is about 10,000 parts per million. In there it's 2 parts per million because we pump ultra-dry air through there, and it's well below the dew point at -78. And then the final important thing it does, although it will pick these samples quickly, which is important to supply researchers with their samples in a timely manner, it does it with complete accuracy. You remember the barcodes on the bottom of those tubes. Anybody who's done this at any sort of scale, well, you know if you try and do this manually, you can get up to a 10% error rate. Now, in your control group, that may not be the end of the world, but in your case group, that is effectively a false positive. So this will do it with complete accuracy, and it will do it in the cold. It will do it at -20, so the other samples are not affected. This is our backup site. I say it's about 8 miles away. Just to show you this, the samples are held in these manual nitrogen vapor tanks. There are about 50 of these. There are about 5 million samples. And you can see there are the monitors and the alarms and the remote filling capability. I mentioned the point about the capacity with centralized archives to invest in infrastructure that will protect the samples. You can see around the wall here these red tanks. These are fire suppression systems. So if there is a fire, this will get triggered and put it out. And then finally, we need to think about issuing these samples out to researchers and also to pool them for the in-house data generation activities that you're going to hear about, the biomarker analysis and the genotyping. And I think it's fair to say, when we designed the big -80 store, we certainly hadn't anticipated we would be we'd certainly anticipated we'd be supporting researcher access requests, but we hadn't anticipated we'd be pulling 4 million tubes for the biomarker analysis and 500,000 tubes for the genotyping. So you're in danger of creating a single point of failure. So what we've done is we've put in place modular capacity to be able to pick these samples and distribute them. So at the top left, this is a smaller modular store that will hold about 1 million samples, but it has a picking station in there, so it can pick these tubes at a high rate and generate orders. You can even go down a level where the center picture just shows you just a picking station. There's no storage capacity there. And then at the bottom, this is the picking station of the very large store that I showed you, which will pick either individual racks or individual tubes. And that space in there is at -20. So I think it's fair to say that all of that upfront thinking and the investment of time by the group led by Paul, that value is now starting to be realized. These samples are currently being used for in-house data generation and also for researchers, and you'll hear about some of these later. And I would encourage you to make applications to use these with the process that Naomi described. Thank you.
think we're fine for some questions. If there are questions to Tim on the samples. Yeah, there's a question here at the front, Andrew. Thank you.
Thank you very much. It's very impressive. I noticed that the size of your aliquot is quite large, so 850 microliters. I mean, you're aware that for some of the study we need much smaller. So what's going to be your approach if we only request, let's say, 50 microliters? Do you consider freeze-thaw, or you will tell us, no, your study is not?
No, I don't think we would take that. I mean, Naomi describes some of the approach. We are certainly looking at ways that we can use the samples efficiently, but we would not do that. We would provide 50 microliters. If it went through that process and it was approved, we would provide 50 microliters. Okay. You would tag it in our information systems that sample had been thawed once. Okay, great. Thank you.
Tim, as a follow-up to that, I mean, if samples are pulled out, will they then be sub-aliquoted at the same time?
Yes, I think they can be. It's interesting to talk about how technology has changed and the smaller amount of volumes. If you come back to the example of metabolomics and talking about the lithium heparin plasma sample, at the time the volume required there was about 350 microliters. That's come right down now. We were also considering one of the very expensive parts of this is the consumables. And the more plastic you have in there, you're taking up more of the storage space. So inevitably, as part of this future-proofing, it was something of a trade-off between having sufficient aliquots that we can balance this sort of freeze-thaw aspect, trying to anticipate the sorts of volumes that people would use in their studies, and not using so many consumables you take up storage space and increase the cost excessively.
So a question over there.
It's just a question of a different kind. I was wondering whether people who are asking questions could say who they are and where they're from.
Yes, who are you and where are you from?
Thank you, Hugh. It's always good to have to be a fall guy. Yes, I'm John Galloway from UCL.
Thanks, John. Any other questions or comments? If not, then there's coffee upstairs. Could I please ask you to be back in time for us to start at 15:15, please? Thanks very much.